Okay, let's get going. All right, I'll end up putting this one on the Monday lecture, and then what I'll do is later on today, I'll go ahead and put another one, and that will be today's lecture. Okay? And yeah, now we'll go start 2 15-minute lectures that we should have had this week. Okay, so the oxidation by the citric acid cycle in the electron transport chain, the pyruvate can be metabolized under aerobic conditions. Everybody's comfortable with aerobic and anaerobic? Okay, fantastic. Through the citric acid cycle and the electron transport chain. Now, I'm going to not end up doing this every time because it's just hard to catch every time. But when I say citric acid cycle and I say electron transport chain, those are not actually two different things, okay? They are subdivided so you can clarify the cycle for them, but they're two halves of the same cycle. It's like me describing the front of a penny and the back of a penny. They are not a different object, right? Okay, so when I say one, understand that I mean both, don't I? They can't actually be split from each other. They are not actually separate things. They depend on each other and happen side by side together, okay? In lockstep. Okay, does that make sense? Okay. So when I say citric acid cycle, what does your brain say? Exactly, it says both, right? Okay, thank you. So this is going to form CO2, H2O. It'll form a great deal of energy. The vast majority of your energy from your metabolism is from this cycle, okay? Which is pretty interesting because it only has 2 carbons going in at each time, right? So the transfer of electrons to O2, this is also happening in the citric acid cycle / electron transport chain. Specifically, it will be accounted for in the electron transport chain part. This is actually also what regenerates your NAD+ when you're going through your normal cycle. Remember, one of the most important things is we need to regenerate the NAD+ so it can go back into glycolysis, right? Because we would lose all of our energy if we had to resynthesize it every time. It wouldn't make any damn sense. So the pyruvate dehydrogenase complex, this catalyzes the oxidative decarboxylation of the pyruvate to the acetyl coenzyme A. Again, this is going to be acetyl-CoA, is the other way of saying that. This is going to do that in the mitochondria, which is why the mitochondria is considered the quote-unquote powerhouse of the cell, right? It's the subdivision that actually does this process. So the pyruvate plus NAD plus coenzyme A, this yields acetyl-CoA, CO2, and NADH plus NH plus. Note this is typically considered as a pre-reaction before the citric acid cycle, okay? So this will be the one that gets you into it eventually. The obligate anaerobes, are going to be organisms that cannot survive in the presence of O2. Some of the examples are going to be the bacterium Clostridium perfringens, this is actually what causes gangrene, and this is why gangrene is typically not something that would be developed in a open wound. Typically that's going to happen in closed off wounds that oxygen can't get to easily, right? Facultative anaerobes, these are going to be organisms that metabolize glucose aerobically when O2 is present and perform fermentation when O2 is absent. Basically, they can do both ways. They're a bit like us, but instead of doing the lactic acid pathway, they do the fermentation pathway. Microorganisms are capable of generating a wide array of molecules as endpoints of fermentation. These are some different examples of pathogenic obligate anaerobes. Again, obligate anaerobes are which ones again? Cannot survive in CO2, or O2, you got it. This is going to be like tetanus, which typically the infection of in a human is what would normally be called lockjaw, okay? Or botulism, botulism. Yeah, kinda. This is the very, very severe food poisoning. Did you guys know that this used to be the leading cause of death in the world wide for all humans for almost all of history? You guys know that? Yeah. It's until the FDA and the food purification for refrigeration came along. Yeah. The only times that wasn't true was even during wars, it was typically true for men. But occasionally women would get a higher rate of dying from childbirth. So it depends on the conditions. This is cat scratch fever, by the way. You get flu-like symptoms. Cats are just wonderful little bacterial petri dishes. This is blood infections and such. What do all these have in common again? If they have exposure to oxygen, would you develop those? No. If you have good exposure to oxygen, you do not develop those typically. So this is the starting and end points of various fermentations. This is glucose. It goes to lactate. Lactate goes to acetate. Glucose can go to ethanol. Ethanol can go to acetate. Arginine goes to carbon dioxide. Pyrimidines go to carbon dioxide. Purines formate. Ethylene glycol, acetate, threonine, propionate, leucine, you get the point. Now, notice though that some of these fermentations, you can actually go on from that fermentation endpoint into a new fermentation. Can everybody say it okay? For example, lactate. Lactate can go to acetate. The fructose is found in table sugar and high fructose corn syrup. To be metabolized, fructose must be converted into metabolite of glucose. So the fructose 1-phosphate pathway metabolizes fructose in the liver, and that actually is going to then go on to form a, right here, the fructose 6-phosphate. Remember, that is part of the glycolysis pathway. Okay, this is where galactose comes in, by the way. Galactose actually has to go to the glucose 6-phosphate, and then that enters the glycolysis pathway. These transformations do require some degree of energy and some effort, which is typically why those are considered like harder to metabolize. Okay? This is actually where fructose comes in case you're curious, all the way down here as DHAP and GAP. Yeah. And you can actually kind of catch this in the liver, this does this. In the adipose tissue, the fructose can come in as fructose 6-phosphate. And again, that's that fructose 1,6-biphosphate would have went to those. The fructose is going to be reacted by fructokinase. This is going to use ATP going to ADP. And so does fructose give you as much energy as eating a similar amount of glucose? Good question, huh? Okay. Anybody remember what happened up here? We had to do what? We had to put in 2 ATPs, didn't we? Yeah, so actually, you're not actually losing that much energy here, okay? You would have actually had to put in ATP anyways. And it's something that people commonly lose track of. They forget that the glycolysis, you had to put in ATP as well, didn't you? You were investing ATP. So again, your ATP goes to ADP, you get fructose 1-phosphate. That's not fructose 6-phosphate, is it? And why am I saying that? Be very, very careful on these alternative pathways. There's a lot of compounds that are very similar. Fructose 1,6-phosphate, fructose 6-phosphate. Those are different compounds, okay? They cannot substitute for each other. So fructose 1-phosphate aldolase is going to be the enzyme that's going to react with this substrate. And this is going to go to glyceraldehyde and dihydroxyphosphate. The glyceraldehyde is going to react using the triose kinase. This is going to convert ATP to ADP. Again, we're going to use up another ATP. And this is going to give us our glyceraldehyde 3-phosphate. What do we notice about those yellow with red borders? What do they look like? Yeah, they look like these, right? And that's because they are those. Yeah. Dihydroxy. Remember that? Okay. And then glyceraldehyde 3-phosphate. Okay. Now, epidemiological and clinical studies have previously linked excessive consumption of fructose to fatty liver, insulin insensitivity, obesity, and type 2 diabetes. This is not supported by recent meta-analysis, and so this very well, in fact, probably is a bad study. Yeah. It not being supported by meta-analysis is a big red flag. Okay. But there was a link at one point in a study. Make of that what you will. Okay? Probably need to rerun the study with a much larger population, and that would help a lot. Overconsumption of sugars and fats is strongly associated with these clinical outcomes. Excess pyruvate is converted into acetyl-CoA and then into fatty acids. An interesting little side note to this is, do you know what actually is linked more to the development of obesity or heart disease? Eating excess protein, fats, or carbs? Which one do you think is more strongly linked? I'll give you a hint. Northern Europeans eat a ton of fat. Way more than the average American. Okay? I mean, Italians eat more fat in the form of olive oil than the average American, right? We actually don't eat that much fat compared to some cultures. What do we eat a lot of, though? What is all around us all the time? Sugar. Yeah, lots and lots of sugar, right? And that actually is the food pyramid. Do you know what the food pyramid is actually designed for? I'll give you a hint. They finally got rid of the food pyramid at the same time they introduced putting ethanol into cars. That's made from corn. So it was literally, the food pyramid was designed to support consumption of, what's the bottom one? Carbs, corn, right? Because we had to figure out what the hell to do with all the corn. It had nothing to do with health outcomes. Have you guys seen the new standards put out by the government? Just the last, actually, it happened right after Biden. Biden's actually the one who did it. Well, I'll tell you what. Eggs has a health score of 30. Lucky Charms has a health score of 70. Higher is better. They went back to the food pyramid. Yeah. So anyways, overconsumption of sugars and fats is the most strongly linked to these health outcomes of diabetes, obesity, insulin insensitivity. Basically, it's a very much linked to the obesity that links back. Okay? More exercise will actually link the other way. The excess pyruvate, again, is going to be converted into the acetyl-CoA and then fatty acids. The galactose-glucose interconversion pathway, which is a 4-step pathway, converts galactose to glucose 6-phosphate. Step 1 is going to be the phosphorylation of galactose by galactokinase. And remember our galactokinase, we're transferring a phosphate on there onto galactose, right? Okay, and that will give us our galactose 1-phosphate. Everybody see that okay? Fantastic. So the glucose is actually going to be converted into UDP glucose, the uridine diphosphate glucose, which is the UDP, uridine D diphosphate, and it's linked onto the glucose, which we see right here. Okay. is going to be that activated intermediate in your synthesis of most carbohydrates. And again, here we have the UDP glucose. Here's the galactose 1-phosphate. The enzyme is going to be galactose 1-phosphate uridyl transferase. What does the enzyme tell you about? We're transferring what's? Uridyl onto the galactose 1-phosphate. See that? Okay. And so we're going to transfer that uridine or uridyl is the group being transferred. And that's the uridine there, has 1-phosphate. It's going to link onto the galactose with the phosphate on it. This will be called UDP galactose, even though the other phosphate technically was attached to the galactose first, right? It still would be UDP here. This is going to leave you here with the glucose 1-phosphate. Notice that glucose 1-phosphate here, that can go in our pathways back over here. Okay, and our glycolysis, if I can ever get back up to the glycolysis. Okay, and so this right here is going to be UDP galactose for epimerase. This is actually going to be reacting the substrate UDP galactose on the 4th position of that sugar. See that? And we're putting an OH on there. And this is going to give us UDP glucose. What do you notice about UDP glucose? It's one of our substrates, isn't it? Yeah. And so this is a cycle. It's a little mini cycle, which will keep cycling out glucose 1-phosphates. Those will be what will eventually plug into the glycolysis. Okay? This UDP glucose is just the previous galactoses being fed in here. Okay. All right, so the sum of the reactions for that galactose-glucose conversion pathway is galactose plus ATP goes to glucose 1-phosphate plus ADP plus H+. UDP glucose is not consumed in that conversion of galactose into glucose. More correctly said, technically the UDP glucose is consumed, but then regenerated, right, by the galactose conversion. Okay? Okay, so the phosphoglucomutase is going to be what isomerases the glucose 1-phosphate to the glucose 6-phosphate that actually gets plugged into the glycolysis. Okay, so this is going to feed into the glycolysis here. That's the glucose 1-phosphate from right there. And again, that's a phosphoglucomutase, mutase meaning rearrangement, right? We're rearranging the phosphate. The galactosemias, these are going to be rare disorders that interfere with the metabolism of galactose. This causes vomiting and diarrhea after things like milk consumption, enlargement of the liver, jaundice, cataracts, lethargy, and delayed neurological development. Often results from an inherited deficiency in the galactose 1-phosphate uridyl transferase activity, which is again up here. The treatment is to remove galactose and lactose from your diet. And so you remove those and they can continue progressing relatively normally along their developmental cycle. This is actually the cataracts right here. Everybody see the cataract okay? Does it always look like that, by the way? No. Does anybody know why it looks like that right now? You see the light here? That's why they check your eye by shining the light into it. If you get the right diffraction angle, the cataract will diffract differently. Instead of diffracting like this, it will become cloudy because it breaks up the light like a crystal. Okay, same sort of reasons actually. So again, your cataract is going to be the clouding of the lens of the eye. This is due to pathological protein aggregation. This is an age disease, but this is also a developmental disease as well. But basically, almost all humans are going to get cataracts at some point. It's a deficiency in our eye's design. Okay, just like our lenses will stop being so malleable at some point. The cataract formation is actually due to this right here. So the aldose reductase catalyzes the reduction of the galactose to galactitol in the lens when the transferase is not active. Galactitol is poorly metabolized and tends to accumulate. Water diffuses into the lens to maintain the osmotic balance. This triggers the formation of your cataracts. And so here's your galactose, here's the aldose reductase, and there's the galactitol. Notice the galactitol has an alcohol there instead. See that? The os, on the other hand, had an aldehyde. Okay? Okay. And so, again, this galactitol, this is going to metabolize very poorly, meaning what? It doesn't get taken out and recycled, does it? Okay? And so it builds up in the eye. What happens if you have something that can dissolve water build up in a place? Anybody remember osmosis? Only thing that mattered was the concentration of your particles, right? You increase the number of particles, it draws more water in, doesn't it? Yeah. And so you're going to draw water into the eye. This is going to eventually trigger the formation of your cataract. And so this is why basically all humans always end up getting cataracts at some point if you live long enough. Okay? It's almost inevitability. I think it actually probably is an inevitability, but not everybody lives long enough. Yeah, I guess that's not really funny, sorry. But gotta laugh if things can't change, right? Okay. Lactase, that's an enzyme that cleaves the lactose into glucose and galactose. So right here we have lactose. This is going to be cleaved through a hydrolysis mechanism by lactase. This is going to give you your galactose and your glucose. The lactose intolerance is the most commonly caused by a deficiency of the lactase. Typically, you do not make the lactase in enough quantity, or your lactase is less active than it should be. And you all have heard of lactose intolerance, probably? Okay. Maybe some of you have lactose intolerance to some degree. It's a very much sliding scale, because you can have an active form of lactase, and yet it's not active enough, right? By the way, is any adult human completely 100% no lactose intolerance? Not typically, no. It would be incredibly unusual. That's why you can't chug a gallon of milk. Okay? You have some degree of intolerance because eventually your enzymes get overwhelmed. The treatment's going to involve the avoiding lactose products or ingesting lactase with milk products. And yeah, you can actually ingest the enzymes. Did you guys know that they also make that for the ethanol as well? As a hangover cure? It's sold in Japan, and it's supposedly now legal in the US. I don't know if anybody sells it. Where they will give you a pill with the enzyme, the dehydrogenation enzyme that we had talked about. Anyways. Okay. You're all like, Dr. Clausen, this is Bethel University. Don't talk about alcohol. Naughty, Dr. Clausen. So your lactose is going to be a good energy source for microorganisms in the colon, which will ferment it to lactic acid while generating methane, CH4, and hydrogen gas, H2. This will cause gut distension and flatulence. Lactate and undigested lactose are going to be osmotically active. They'll draw water into the intestine. This will cause diarrhea. And so this is why lactose intolerance typically causes those side effects. Okay? The enzymes catalyzing the irreversible reactions in metabolic pathways are control sites. These are controlled by allosteric effectors, covalent modification, or by regulating transcription. You probably were wondering why I taught you that just massive library of terms, right? This is how we control this and that. That's so I could do that. I know, I've referenced almost every chapter within the last 10 slides, haven't I? Yeah, we build up, don't we? And surprisingly, you actually knew what I just said, didn't you? Yeah, pretty good, huh? So your glycolysis, this is 3 enzymes that catalyze irreversible reactions. We have hexokinase, phosphofructokinase, and pyruvate kinase. What do you notice about all those? They all end in kinase. Why do they all end in kinase? Transferring a phosphate, yeah. And so we have a huge energy start, and it pushes that reaction forward, doesn't it? Okay. So glycolysis in skeletal muscles provides ATP primarily to power contraction. The primary control of muscle glycolysis is going to be the ratio of ATP to AMP. High levels of ATP allosterically inhibit the phosphofructokinase. Again, phosphofructokinase being one of the committed irreversible reaction steps, right? And so these are the ones that are typically going to be your control points. Okay? This right here being a feedback inhibition that can occur, or a feedback control, right? And so high levels of ATP are going to allosterically inhibit phosphofructokinase. Binding to a specific regulatory site is going to lower the enzyme's affinity for F6P. AMP is going to reverse the inhibitory reaction of ATP. Why does AMP reverse that? If we have a lot of AMP versus ATP, what does that mean? Exactly. Yeah, that's like that ratio of ATP to AMP. It's like our battery charge, right, for our cells. Okay. When we're low on ATP, high on AMP, it means we're discharged. Now, our cells don't normally like to get below 90%. That's pretty rare, actually. That means we really drained it, which is pretty high if you think about it, but whatever. The enzyme activity increases when the ATP-AMP ratio is lowered. Your committed step is going to be the first irreversible reaction unique to the glycolytic pathway. So the example would be phosphorylation of the F6P to F16BP by phosphofructokinase. This right here is your structure for your phosphofructokinase. Notice the different colorings here. Again, those are those different sub, right? And this right here is going to be your allosteric sites. What do you notice about this sort of structure again? Anybody remember our hemoglobin? Way at the beginning? Yeah. So one of the reasons you started on that is because it's something fairly easy to understand because you can visualize it. You actually see this process happening in your body. But, red versus blue. And it has this same sort of allosteric control site reactions that occur in a lot of these things, okay? And so we actually have very similar patterns here that show up all over the place, right? These right here are catalytic sites. Right there. There's 4 of them. And so you have 4 catalytic sites. Okay, what are the allosteric sites again? Those control. Yeah, they're going to control catalytic activity while not being the binding site or active site, right? Yeah. Okay, notice that the catalytic sites are different than the allosteric sites. Why are these red things in those allosteric sites? Yeah, notice the 2 here, those being like the phosphates, right? Yeah. So this would be where you'd have the AMP versus ATP. Yeah. Notice this one has phosphates on it. So that's probably your ATP, right? Okay. By the way, there's 4 of these allosteric sites. Can you guys see these other ones that are hidden behind? Okay, so the high ATP is going to convert the hyperbolic binding curve of F6P into a sigmoidal one. So this is your high ATP. And again, this would be your reaction velocity versus your fructose 6-phosphate concentration. And so when you have low ATP, this is going to tail off really fast here. So your reaction velocity is going to pick up and tail off very quickly. So you get almost maximum right away, right? And you're really going on that. This right here is then going to be the sigmoidal curve. And notice again, you need a lot of fructose 6-phosphate to push that through, right? To get up near the max. Okay? Okay, so a decrease in the pH is going to inhibit the phosphofructokinase activity by augmenting the inhibitory effect of your ATP. This is going to occur when the muscle is functioning anaerobically, and it's going to produce excessive lactic acid. This will prevent muscle damage from too much acid. So what ends up happening is when you're going anaerobically, right, then you're using up a bunch of your ATP, aren't you? But you have a problem. If you just kept going and building up acid in there, what's true about all biologically active compounds? If they're in the right concentration, they're good, right? What if they're too high concentration? All medicines are poisons. All poisons are medicines, right? All substrates in the right concentration are great reactive ingredients, right? Substrates in the wrong concentration, too high concentration, end up being what? Toxins. Do you remember fermentation and how I said it kills itself off eventually? And that usually happens in any closed environment. Okay? Guess what happens in a closed environment in our body if we can't keep up with lactate? You could kill yourself off, right? Kill those muscles, permanently damage them, building up too high of a acidic concentration. And so what ends up happening is you actually have a feedback effect where this is actually going to slow this activity if you start getting too acidic. Okay? And it's just going to peter out, run out of your ability to produce any more energy. Right? And your body's going to go, no. No more. We're done. The ADP, this is not positive regulator of phosphofructokinase because the adenylate kinase is going to form ATP from ADP. And this is why you may have been wondering all this time, like, why is it always AMP? What about ADP? This is why. Okay? So when you have two ADPs, you can actually assemble them into an ATP and an AMP. And so you can recycle and recover that energy, right? And remember, what's your energetically most favorable step in these things? Is it taking both phosphates off? Do you get the most energy from that single reaction? No, you get the most energy, but it's not energetically the most efficient, right? Yeah, you only get 50% of that extra phosphate. So the AMP is going to be the one that serves as that signal for your low energy state. The AMP is going to be an allosteric regulator. This will provide sensitive control because, again, in the cell, ATP is greater than ADP, which is greater than AMP. Okay? And so AMP is going to be your least concentration one, typically in a cell. And your ADP is going to have a typically higher concentration. So small changes in your ATP are going to result in large changes in AMP. So if you have a huge increase in your ATP usage, you're going to generate a lot more AMP fairly quickly. It'll be a more sensitive change. Let's put it this way. A 50% difference in ATP concentration is a big difference in concentration, right? You have to use a lot of ATPs. A 50% change in AMP is a lot less AMPs, right? That makes sense? So the hexokinase, is this going to be inhibited by its product, which is G6P. The concentration of G6P signals that the cell no longer requires glucose for energy or the synthesis of glycogen. This is going to lead to your glucose remaining in the blood. The inhibition of the phosphofructokinase is going to lead to the inhibition of the hexokinase because a rise in F6P leads to a rise in G6P as they are in equilibrium. The pyruvate kinase is going to allosterically inhibit by ATP. This slows glycolysis when energy charge is high. Feed-forward stimulation or feed-forward activation, both of those terms being mostly synonymous, is going to occur when the products of a preceding irreversible step activates the enzyme. So one of those examples is going to be when the pace of glycolysis increases, the F16BP is going to activate the kinase. This right here actually shows these inhibitions and activations on the pathway. So this is at rest. Your glycolysis is going to be inhibited because you're not using up energy really fast. Okay? So you can do this relatively slow just to replace the phosphates you need, your ATP you need. And what you're seeing here is this is the negative feedback loops. So the glucose 6-phosphate, this is the product. It's going to come back and it's going to inhibit hexokinase. The ATP being high versus AMP, i.e. your high energy charge, this is going to inhibit PFK and pyruvate kinase, both of these right here. That's the one PFK, by the way, that goes fructose 6-phosphate to fructose 1,6-biphosphate. And this pyruvate kinase being the one that does the phosphoenolpyruvate to the pyruvate. Again, this would be your relaxed muscle fiber. You're sitting around listening to Dr. Clausen, hopefully, because you have nothing else to do in life, of course. During exercise, this is going to be glycolysis stimulated. So what's going to happen is you're going to have a low energy charge. You'll have used up some ATP, and you'll have more AMP now. This is going to activate the PFK so that we get a greater activity. And so the fructose 6-phosphate will convert faster to fructose 1,6-biphosphate. This will also have feed-forward stimulation. That fructose 1,6-biphosphate actually activates the pyruvate kinase right down here. And this is going to cause the phosphoenolpyruvate to be converted to pyruvate faster. Okay? This will be your muscle fibers are contracting and doing their jobs, right? Working out. Okay? And this one, again, is still listening to Dr. Clausen, but working out while you're listening so you can maintain your health, right? So you're ready for those exams. Okay? Because, again, listening to Dr. Clausen is all that exists in your lives, of course. Okay. Now, the regulation of glycolysis in your liver is going to illustrate that biochemical versatility of the liver. So your liver maintains your blood glucose concentration. This is going to generate, reducing power for biosynthesis, and it's going to synthesize biochemicals. Regulation is actually more complex than in your muscle because it has to have a more fine-tuned approach because, again, it's going to do this basically be your secondary concentration reservoir and change for your body, right? Okay. Do you guys know where the glycogen is stored, by the way? If somebody drinks a lot of beer and eats a lot of carbohydrates, where do they get fat? The gut? Anything located around there? The liver? Yeah. This is why that happens, is because the liver is doing the heavy lifting for this. Okay? Your liver phosphofructokinase, this can be regulated by ATP, not an important metabolic signal, as your liver does not experience sudden ATP needs. Yes, it still exists, that still is there, but how often do you exercise your liver? Don't admit to it. There's a secondary meaning to that. But how often do you lift weights with your liver? Not often, right? It's not a muscle. So, yeah, that's not happening much, is it? Liver phosphofructokinase, this can be regulated by pH. Again, it's not an important metabolic signal. Your liver does not produce lactate. It does have some pathways to move things around, but again, it is not your muscle, is it? It is not going to end up anaerobic where you're working out too fast and you can't keep up. It is not going to end up using up a bunch of ATP because you're lifting muscles or thinking really hard. It's not doing any of that, is it? Yeah. The phosphofructokinase, this is going to be inhibited by citrate. This is going to be an early intermediate in your citric acid cycle by enhancing the inhibitory effect of your ATP. The high citrate actually means the biosynthetic precursors are very abundant. And so this is a fairly useful indicator for your liver because your liver actually does quite a bit of biosynthesis. The phosphofructokinase is going to be activated by fructose 2,6-biphosphate. That's the F26BP. Binding of the F26BP is going to increase the affinity of the enzyme for the F6P and diminish the inhibitory effect of your ATP. And this actually summarizes that right here, in case you're wondering. And so that's going to be activating that PFK. The high F6P, this is going to accelerate the synthesis of F26BP. And again, that's that F26BP, in case you forgot. Didn't know what was going on there. Okay, and so this is going to be some of the different activation curves that we've seen. This is going to be the one for the F26-biphosphate. This is your relative rate of your fructose 1,6-biphosphate synthesis, and this is 100 right here. This would be the 0 to 100 right there. This is the fructose 6-phosphate right here. Concentration, again, the more substrate you have, the more active things will be able to be because we have more substrate to use, right? This right here would be your different concentrations being put in. And what we can see is 0.1 micromole here, micromolar, is actually quite a bit of activation property, right? When we get up to 1 micromolar, this thing skyrockets almost immediately to max. Everybody see that? It's quite effective, right? This right here would be your relative rate of your fructose 1,6-biphosphate synthesis, and here would be your ATP concentration. This one being in millimolar as well. And again, this right here would be 0 concentration of the 26BP, the F26BP. This would be 0.1. And this one right here would be 1 micromolar. And again, what can we see here is that, again, we get almost all of this activity here is front-loaded for your ATP. But as we go out and get more of this, the ATP is going to be less and less effective at blocking this, isn't it? And so we're going to have more and more activity even though we have higher concentration of ATP. Though keep in mind that eventually this does have an effect. Okay? Having a high enough concentration is going to slow this down eventually, even at 1 micromolar. Though not to 0, you notice. Not anywhere close to 0. Okay, the hexokinase is going to be inhibited by its products. That's G6P, as in the muscle. That's that same inhibition we had saw in the muscle. The glucokinase is going to be a specialized isozyme of the hexokinase. This can provide the G6P for glycogen synthesis, and fatty acid formation. It's not inhibited by G6P. The functions as a monomer, but it actually displays sigmoidal kinetics, which is typically not what we would see for a monomer, right? That's usually what we have when we have multiple subunits that are interacting with each other, right? The phosphorylates the glucose only when glucose is abundant, inhibited by the liver-specific glucokinase regulatory protein, that is the GKRP, when glucose is low. Several isozymic forms of the pyruvate kinase are going to be present in mammals. We'll have the L type. This is predominantly in the liver. I know the naming here is wonderful, isn't it? You could tell that the chemists have had an influence on those biochemists. Because otherwise it would have been like somebody's name type, right? But no, liver, L type. Okay? M type, guess what that is? Muscle, yeah. Now, to be fair, it's predominantly in the muscle and the brain, and you can have mixtures depending on the tissues. Okay? It's not necessarily all of one or the other. But brain is, you can think of brain as kind of like a muscle, right? We have a lot of activity, a lot of heat dissipation, a lot of energy being used. Huh? Yeah, unfortunately. Kind of like everything in organic chemistry is German and French. Yeah. Yeah. Most of biochemistry happened when English was the predominant language of science. And so there's still the same abbreviations in every language, even in Chinese and Egypt, where they don't have these letters at all. Still M and L. Which is actually amazingly impressive, actually. But, yeah. The L forms, this is controlled by reversible phosphorylation. By the way, it is trippy to read papers in Japanese and Chinese and stuff and be able to mostly understand them, or German or Italian, French, and mostly be able to understand like every third letter, every third word. Yeah. So anyways, the pyruvate kinase is, did I answer your question? No problem. No problem at all. Yeah, basically what ends up happening if you go into other languages, is you end up with the exact same abbreviations. Yeah. They just use those same abbreviations from the discoverer. And what you do is you put the connective words in your own language. Yeah. So it would still be L, and then you'd use type in your own language. Which is kind of weird, but it is what it is, right? And the 0 through 10 numerology that we're used to also typically gets used, even though the number of these languages don't have that. Yeah. So the L form, this is going to be controlled by reversible phosphorylation. When blood glucose level is low, the glucagon-triggered cyclic AMP cascade is going to lead to the phosphorylation of pyruvate kinase, which diminishes its activity. And you can start seeing where these are connecting to our other pathways. Remember that? Where we talked about the receptors and glucagon reception and how that changed the kinase activity through phosphorylation and did that cascade. Okay? I'm going to be honest, there is no way you're going to be able to make all of the connections in this semester. I'm going to give you the language, I'm going to give you the concepts, and I'm going to try to make it so you understand these connections can exist and point out some. Okay? And I'll give you that web, and that web actually has most of those connections kind of made. But you're going to be like the next 10 years, if you're in anything related to this, and be like, oh shit. Okay? It'll take a while to process. So again, this is that pyruvate kinase activity. This is how it's controlled by the phosphorylation dephosphorylation that we mentioned before. So this is low blood glucose concentrations. Here we're going to have that dephosphorylated pyruvate kinase. That's the more active form. I know that it's hard to keep track of. This is why you get these repeated. So we're going to have ATP going to ADP. This is going to phosphorylate that. Notice the P. We phosphorylated that. And that's the phosphorylated pyruvate kinase. In this case, that's the less active form. Please don't get that in your head that phosphorylation means less active. Okay? In this case, yes. Okay. High blood glucose concentration. This is going to cause that to be hydrolyzed. Notice it does not go backwards in the reaction. Does not get that ADP going to ATP. Okay? It's not actually resetting the reaction completely. That will dephosphorylate that and give us the dephosphorylated pyruvate kinase. This is the more active form. And I guess I will end here. Can I finish this last little statement? Or? Okay. So that's that reaction. This is the enzyme that acts on phosphoenol pyruvate, ADP, going to pyruvate and ATP at the end of that. And this is this one right here, where we have the more active form, fructose 1,6-biphosphate, and then ATP and alanine will cause that to become the less active form. Okay? There we go. Anybody have any questions? Okay. You have a wonderful day. I will see you tomorrow on Thursday. I'll try to release a video sometime this afternoon to catch us up on our lecturing, okay? Mm-hmm. Oh, yeah, I'm just giving you 90% of that in terms of, I've actually given you a bit of extra points than I would normally give on a review assignment like that. Okay. You may notice instead of it being 5 questions for 1 normal point, it's a better ratio. Yeah.
So if we don't do like all the questions, we do some of them, do we get like partial points?
You do? Yep, because it's out of 0. Yeah. So whatever points you do get,